Are we ready? No. Who's ready? Uh-oh. Okay, so today was our first recitation meeting. I think it went okay. What do you think? Everyone feel good about it? Okay. The room is weird. Actually, I think one section is in 265 and everything else is in 221. And I don't know how I feel about that room, so I was stalking all the other rooms to see about changing, but I don't think we're in any luck in that. So we'll see what's happening. So we're going to have to talk about how we're going to sit so we can work best together without large spaces between us. So we'll see how that works. Okay, so today we're going to continue the methods that we started the other day, and we're going to talk about primitive structure and function, which you guys already taught yourself about through the quiz this weekend. First, a little bit of business. Please see me after class if A, you have not been added to Blackboard yet, and B, you just were added and still need to take the pre-test and quiz 1 because you didn't have access before and you missed it. So come see me because I want to know who's where on what, and what I'll end up doing is opening up those other two assessments once everyone has access. And then I'll just email all of you and say, hey, it's open for this many hours, do it. Okay. Stump the teacher. So the other day I was asked a question about signaling and the effect of glycosylation on signaling through a receptor. And I didn't do a good job answering it, so it became a stump the teacher question. Lots of people participated, yay. The answer pretty much was glycosylation can have an effect on signaling, which isn't much of an answer. So that pretty much was the answer. And I'm not satisfied with it, but we're going to just stop with that because it's not 100% what we're talking about in this class anyways, and I'd have to dig deeper and waste class time, but it was a very interesting question. Those of you who participated will get points, well, a point. Normally I don't just give points for participating, you actually have to get it correct, which requires you to answer the question. But I usually start out by just, I give you your freebie. In the future, some tips on how to answer a question. Make sure you know the question, because a lot of people answer questions that weren't asked. So just answering a question when it's not the question being asked, you'll, in the future, not get points. So confirm with a classmate. Be like, I'm not sure if I heard that right. And then the other thing is answering by restating the question is not a way to answer. So just, and this one was a hard one because really the answer was kind of like, sometimes there are different alterations in the pattern of glycosylation which can affect the specificity of the receptor for the signaling molecule, which is really a lot of hand waving and nothing really mechanistic. I like mechanism. I want like deep answers, not just like it has an effect on signaling, not an answer. So I think that's something that we just learn about through the semester with me. It's like exactly how much of an answer I want, but it's extra credit, so if you don't get it, it doesn't matter, right? It's not like you're failing an exam. So in the future, I'll try to give some feedback on the types of answers that I get and what is enough and what is not, and you'll start to get a feel for what's enough. But for now, everyone gets a point. Okay. Last time we were talking about the concept of a cell as part of a larger organism that needs to respond to the environment. So you have all sorts of things going on, but the big one is you have signaling that then kind of affects many processes, but one big one that we're focusing on this class is gene expression, and that's pretty much the big picture I want you to keep going back to. And then we started talking about how people study gene expression through northern blots, and we did a nice activity there where we spoke about some actual data and how to form hypotheses and how to analyze data. And then we stopped with real-time PCR. So I'm going to finish that stuff, and then we'll start talking about chromatin structure and function. Okay. What kinds of questions? So I ended with explaining real-time PCR to you guys, and that was just, it's a way to quantify the amount of RNA for a certain gene. So you have primers to the gene of interest, and you convert your total RNA to cDNA, and then use those primers to amplify your gene of interest. And based on the cycle number at which it goes exponential, you can tell how much of the RNA there is relative to, say, a control. So you might say to me, so quantitative assessment of amount of RNA in a cell, right? Because if you're analyzing gene expression, the quantity of RNA tells you something, right? Specific RNA, not total RNA. Lots of confusing looks and faces. Yes?
Can you repeat the definition?
No. qPCR, quantitative PCR, also known as real-time PCR, is quantifying your RNA of interest. And that's how you can assess the effect of whatever treatment on gene expression. So for example, in the lab, we were doing an experiment, and I'm going to actually give you some other stuff, but in the very simple one. In the lab, we were doing an experiment. We were trying to see if treatment with certain drugs was going to increase expression of a gene called colon-5. And so our RNA of interest was colon-5. And so our primers were designed to a region in the colon-5 gene. And so we would have our cells treated and untreated. And then we would isolate all the RNA from the cell, turn it into cDNA, and that's only because cDNA is stable and RNA degrades if you stare at it too long. And then we would do our real-time PCR, and our primers would be specific to our gene of interest, and it would increase, because that's what PCR does, it increases the copies of your template of your gene of interest based on your primers, and then we would be able to decide to figure out how much of that RNA is present. So it tells us, is the expression increased because maybe your RNA levels went up. Does that make sense? Yes, no, maybe so? Yeah. So let's see. If you don't say no, I want you to shout out no when I ask you if it makes sense. Because I'll never judge you. Well, I will. But I'll judge you for not saying you don't understand when you don't understand. So just a few other examples. What other kinds of questions we can answer with real-time PCR? Anytime you want to look at whether or not some gene is upregulated. Upregulated means expressed at a higher level. I use upregulated, downregulated a lot. Upregulated, more expression. Downregulated, less expression, usually in response to whatever stimulus. In this case, we were treating with some drug. So what other kinds of questions can we answer? So another thing that we did in my lab is we are looking at the role of KSHV, which is a virus, Kaposi sarcoma herpes virus, on latent to lytic transition, because it's herpes viruses can be latent and then they can go lytic. And we're specifically looking at one protein and looking at its effect on two different genes. TNF alpha, which is up here on this graph, and ICAM1. And so the question was, can VFLIP activate TNF alpha or ICAM1? And what happens if we express another gene? Will it go down? And so we had our control, which is pcDNA, which is empty vector, so we did nothing to these cells. And then we had VFLIP, which is our viral gene. So just look at those first two. And this is relative TNF alpha expression, so that's just some gene. You can call it gene X if you're getting confused by names of genes, right? And so when we did nothing to our cells, would you say this is low expression? Yes. When we added in VFLIP, would you say there was an increase or a decrease? Increase. An increase, right. And same with ICAM1. When we did nothing, it was low, and when we added a V-flip, it went up. And the big thing we were trying to understand is, well, what happens when we put in this other gene that induces lytic replication? And it actually turns everything off, which is great, but neither here nor there. For this example, what's important to understand is you can do something to some cells, and it'll affect gene expression, and you can quantify that by having primers that are specific to whatever gene you're looking at. So here our primers were to TNF-alpha, and here they were to ICAM1. But in both cases, we extracted all our RNA, turned it into cDNA, and then used those primers to amplify those genes. And based on how much product there is from your PCR, you can quantify your gene expression. Okay? So that's real-time PCR. How do we feel about real-time PCR? Do we love it? I'm very visual, and so when I see people who are like this, then I feel good. When I see people who are like this, then I feel nervous. But when I see people who are just like this, then I feel like I'm speaking in, what language does no one hear now? Togo? Who knows Togo? I could have been right. I don't know. What do I know? Anyways, feels like I'm speaking in Togo. Okay. Anywho. So, So we're going to move on, and you come find me if you are like, oh my god. But just really understand that you can look at gene expression by quantifying RNA. Okay. Microarrays. So this is a way to assess genome-wide gene expression, because microarrays are looking at all distracting. Sorry, whenever people walk in late, I clean it out. And then eventually I lock them out, so it won't happen again, though. Never. Okay. It's so distracting to me, though. I'm very distractible. So, gene expression, microarrays, whole genome, right? So you have, in this case, again, whenever you're looking at gene expression, you need to have a control, which should be some untreated sample, and some treated sample. Because remember, gene expression is affected by the environment, and you're changing the environment by treating your cells a certain way. So your sample could be as simple as cells treated with a drug and cells not treated with a drug, a mouse infected with malaria, a mouse not infected with malaria, whatever you want to do. And so you'll have control sample 1 and experiment sample 2. And so again, it's the same thing where you extract all your RNA and you do reverse transcription to make cDNA, and you actually label your cDNA with different colors. Your control DNA will be labeled, in this case, green, and your experimental cDNA will be labeled, in this case, red. And then you'll incubate it with a chip. And it's literally like a glass slide that's spotted with different oligos, different DNA sequences that are specific to every gene in the genome. And so it's using the fact that you have this antisense binding. You know what antisense binding means? No. That's, base pairing, kind of like base pairing. So, you have the base pairing between your oligo on your chip for each gene of the genome and your RNA that you've extracted from your sample. So if there's a lot of it, there'll be binding, and if there's none of it, there will be no binding. And remember, you've made it fluorescent. And so what you end up getting is this hybridization to this chip. So that means the binding of your cDNA to the chip. And if you get a red spot, that means that your gene is expressed higher in your experimental because you labeled your experimental red. If you get a green spot, it means it was expressed higher in your control. And then if you get yellow, it's expressed the same in both. Right, because they're both binding to the same things, and the colors are going to overlap, or one's going to dominate. And so you can start to understand relative gene expression based on that. Yes?
Did you say the antisense binding was binding cDNA to the chip?
cDNA, yeah. So on your chip, so the question is about antisense binding. Think of it as base pairing. And so you're going to have these oligos, like these small DNA fragments, that represents every gene in the genome spotted onto the chip. And then you're going to have your cDNA, and it's going to bind by base, kind of by similar to base pairing. So it's antisense on that oligo. Yeah, so that's how you can look at every single gene in the genome, because you've got all your cDNA coming from your cells, and then on the chip you've got all the oligos in the whole genome. Okay, and so why is this useful? Well, I have my own opinions about microarrays, but it's useful because you can look genome-wide at the effect of something on something, which is useful if you're trying to understand the response to a drug or the response to some kind of infection. Because then you get all this data, and you just need to learn how to deal with the data and find the genes that are most upregulated or most downregulated, and then you go back and do the experiments on those specific genes. Okay, so it's kind of like a fishing expedition in a way, and you find some fish, and then you do further experiments on those fish that you caught. Okay, so you can't actually do microarray and publish a paper. You have to do microarray and figure one. Yes?
Am I right in saying this is slightly limited because you can't have as many little spaces as you have genes?
So you're saying it's limited because you don't have as many spaces as you have genes. Actually, so they have chips that are the whole genome. Yeah, so this picture is just a picture, but in reality that you can buy the human genome on a chip, or you can go specific. Let's say you're just interested in kinases or transcription factors or one viral genome. So you can buy, like there are these companies, you don't make the chip, you buy the chip. And you just say, hey, I want the human genome. And so you can use that. Yeah. But it is limited also because it's really expensive, and you can't just do one, you have to do replicates, and if you have many conditions and many replicates, it gets very expensive. Then you need to analyze all that data. So it's powerful and limited all at the same time. Yes?
Could you just tell us what cDNA is? I'm sorry if you already went into
cDNA. The question is, what is cDNA? Thank you for asking that, because I just make assumptions. You need to stop me on that. cDNA is a copy of your RNA. So it's taking your RNA and using reverse transcriptase to turn it into DNA. And so C is for copy. So it's a copy, a DNA copy of your RNA. And the reason you do that is because RNA is very unstable. And by turning it into DNA, I mean, DNA is the most stable thing in the world. Whenever I see people with DNA on ice, I want to laugh at them. Because you can put it in your pocket and ride cross country, and it will be fine. It doesn't matter. DNA is so stable. You can like people have told me stories about how someone wanted to get a plasmid from another lab, and that lab wouldn't share their reagents. And so they actually sent them a paper, and they took the paper and crumpled it up and put it in buffer in a tube and extracted the plasmid out of the paper, because they knew the paper had been in the lab and that plasmid was everywhere in the lab. So it's so stable. But anyway, that's cDNA. Does that make sense? Okay. Okay, I feel like I'm spending too long on this. Any other questions? Were you raising your hand?
No, it's about the
Okay. Okay, good. So those are microarrays. Microarrays were really hip and hot in early 2000s. I think it's hard because these kind of things need to be reproducible, and there's some issue with reproducibility when you do these very high throughput assays. I guess now it's getting better, but I don't know if it's nearly as hot as it used to be. It was the next big thing. Now everyone's looking at the epigenetic modifications of the whole genome through sequencing. It's always how big can I make my science? Okay. So what kinds of questions can you answer using a microarray? Well, so what I just told you is you can treat different cells. You can treat cells with different drugs. You can infect mice with different things. There's a lot of things you can do like that. This is from a paper where they were looking at breast tumors from different patients to look for poor prognosis gene expression signatures. So you have these tumors. You do microarrays on each 117 microarrays, actually, when you consider the fact that you had probably duplicates at least. That's a very expensive project. And then you look at the whole genome and you can start to see across all these different tumors from all these different people what genes are associated with poor prognosis, right? So all these people who did really poorly, well, they all had really low P53 or really low apoptotic related factors, something like that. And so that actually is really interesting, right? So you can say there's a lot of power to doing a study like this, because then you can start to kind of predict that, you know, if you prove that certain gene expression profiles are an indicator of poor prognosis, then you can start to screen people, right? You come in with cancer, let me take your some cells from your tumor and do some real-time PCR, because you wouldn't do a microarray on the person, but you've already identified genes that are interesting from doing a study like this, and say, well, let me check this gene, let me check this gene, let me check this gene, and then you can predict people's prognosis, you can predict their outcomes, you can design different treatment strategies, because maybe certain genes are associated with response to better drugs, better response to drugs, stuff like that. So this kind of thing, I think it's quite powerful. How are we doing?
So, microarray analysis tells you which gene is being expressed or how much a gene is being expressed.
Microarray analysis doesn't tell you which gene is being expressed as much as it tells you the level of expression relative to other things. Yeah, so you'll have your control and then you'll have your tumors. You would have like for this example, they probably took breast tumor plus adjacent tissue that was normal. So their control was the normal tissue, and then they looked for that change in the breast tumor. Yes?
So is it correct to say that you can use microarrays for screening for genetic disease?
You can use microarrays for looking at gene expression differences. So genetic diseases often have to do with mutations and stuff like that, where then you can use PCRs to amplify specific genes and sequence to look for mutations. If a genetic disease had an outcome that resulted in high or low expression of a certain product, then you could do that. But then usually you don't want a whole microarray because you might know a gene. So it's not as, well, I mean, it might be useful, but it's not the way that people do it. Yeah. Okay, any other questions? That was a good one. Good. Okay. Reporter assays. So all of that had to do with RNA. But before you get RNA, you need transcriptional activation. That usually, you're right, so before, transcription results in making RNA, so you need to activate transcription, right? So that usually involves binding of some proteins to some regions of DNA, often transcription factors binding to some enhancer regions. And you can use a reporter assay to look at this. And so usually what you'll do is you'll have something you can see, such as luciferase, which makes light, firefly luciferase. You can see that when the firefly does its thing. GFP, that's fluorescent. And so what you do is you put that gene under control of whatever your promoter or enhancer of interest is. So let's say you wanted to study activation of that NF-kappa B pathway. Well, it's known where NF-kappa B binds to the DNA to activate that signaling pathway or to activate genes, right? So you can just put that ahead of your luciferase or ahead of your GFP or whatever, and then you do your experiment, and if NF-kappa B is activated, you get a lot of luciferase. So it's a reporter assay. It's because your gene that you can see, luciferase or GFP, is reporting the activity of that transcription factor binding region, or transcription factor. Okay? So I'll give you guys some examples here. So again, you're looking at control, like you're comparing treated. With all this, it's not useful unless you have an untreated control, right? Because you can't look at things being turned on or off, whether or not you know what the baseline is. Okay? So what kinds of questions can you answer using a reporter assay? Basically anything where something's activating transcription from a certain promoter or enhancer. So this is an example of with NF-kappa B. So a major activator of NF-kappa B signaling resulting in NF-kappa B associated genes being expressed is TNF-alpha. It's just a cytokine, binds to receptors, signal, transcription factor to nucleus, bind to DNA, gene expression. And so here what we're asking is the transcription factor binding to the DNA to activate gene expression? And the readout is, in this case, GFP. And so you can see they're treating, so you've got your negative control, and then you've got different amounts of TNF-alpha. And so what you're looking at is here actually is TNF-alpha over 0, 6, 12, and 24 hours. And you can see your green is increasing over time. Might be hard to see for those in the back, but here's 0, 6, 12, 24 hours. Would you say your green is increasing over time? That's actually a microscope looking at the green in cells. And so you've transfected your vector, which contains your GFP, into your cells, and it's being controlled by your NF-kappa B transcription factor. And so you would say TNF-alpha over 24 hours had activated NF-kappa B pathway, which would be expected. And here it's just graphed where they made it relative to untreated. And there's you could just you set untreated to 0, you set and then you can make everything relative to that. So you can ask a lot of questions here. For example, you can see how different pathogens might affect promoter activity. A lot of pathogens affect NF-kappa B signaling. So you can infect with your virus of interest and put your reporter in and see whether or not, compared to uninfected, there's any change in activation of NF-kappa B, for example. Okay? Yes?
So it does help you know if the actual transcription factor is working or not?
Exactly. It tells you whether or not your transcription factor is working or not, whether or not it's turning on gene expression or not. And it's just instead of, so for example, maybe gene X would be activated by NF-kappa B pathway. Instead of having to do a Western blot and look at expression of gene X in response to whatever, you could actually just look at GFP because you put GFP in that place. You can see, is GFP turned on or not? Yeah, but whether or not, I like that, whether or not transcription factor is working or not. And so it's a common way to assess whether a pathway is being activated, whether a signal transduction pathway is being activated. Any questions about this? Yes?
So reporter gene is the same as reporter assay.
The reporter gene, okay, so the question is the reporter gene the same as the reporter assay? So reporter assay is the assay, and then you'll have reporter gene in the assay. So your reporter gene can be GFP, it can be luciferase. There are many types of reporter genes in reporter assays. Make sense? Okay. I love reporter assays because they're just so easy. Like there's so few technical difficulties involved. Is it green or is it not? Okay. There you go. Okay, questions? I think I have, yes. So I also have done this in my lab where we're trying to see whether or not the NF-kappa B pathway was activated by Kaposi's sarcoma herpes virus. And so we did vector, which just means like this is our control, so you have a low luciferase. And when we put in VFLIP, which is this viral gene, what happens? It goes up. And then when we put in our other gene that we're interested in, that we think turns it off, it goes down. And so also, We looked at TNF alpha, which before I said activates NF kappa B, and so it does what it should, it activates NF kappa B. And so then we did some other stuff there too, but I'll keep it simple. And so you actually quantify your luciferase, in this case with a luminometer. It's a machine that can actually read luciferase. Yes?
In this case, the graph is kind of giving you, the relative luciferase units, that's proportional to the amount of NFKB
It's actually really, it's proportional, you make it relative to your untreated. So it's how much higher than your untreated. Yep. And it's a kit that you buy from a company. That's the thing, I have someone new in my lab and they keep saying to me, wow, I thought science was so much more difficult, and it's just all like you just put this and this together and the magic happens. And it seriously is a kit. You're like, take your plasma, put it in, you then lyse your cells, mix it with A and then B and then C and then put it in the machine and it gives you a number. Very surprisingly simple. Okay, that's enough of methods for you guys. Well, for now, until the next recitation. So I want to take this moment to do a public service announcement for people learning how to read primary research papers. So there's going to be a lot of papers you're going to be reading, and I want you guys to be efficient in how you do it. And so here are my tips. 1, don't read the methods, ever. Unless you're trying to recreate the experiment, you don't need to even pay attention to that section. Don't read the discussion, because that's the author saying, look at what I did, it's really important. You don't care what the author thinks, you want to assess the data, right? So don't read the discussion, don't read the methods. Read the intro so you have the gist of the background of what's going on, read the abstract, of course, and then go through the figures. Don't read through the results unless you have to. Look at the figure, read the figure legend, and then look back at the results just to say, okay, what were we doing here? Why were we doing it? Then try to look back and really understand the figure. And if it's a method you don't know, Google it. But that's way faster to look through figures than to like read from beginning to end. So hopefully you guys will give that a try. And it's harder at first and it gets easier over time. Okay. So let's start talking about molecular biology because we haven't really done that yet. We're doing that a little bit. Okay, so we're going to start by talking about chromatin. And a lot of this hopefully will seem very familiar to you because we're starting slow and it's what you read a little bit over the weekend. So, structure and function of chromatin. What does this mean? When I say structure and function of chromatin, what does that mean to you? Yeah? How it's put together and what it does. So the structure. So what do you mean by how what's put together? The chromatin. How the chromatin is put together and what it does. How that affects what it does. Okay, so what is chromatin?
DNA and protein that make up chromosomes.
Right, so DNA and protein make up chromosomes. Right. Okay. So so what kinds of proteins would you expect to find in your chromatin? Yes. Transcription factors. Histones. Those are 2 big ones. What other proteins? Anything else? Those are some big ones. Yep. Huh? Huh? Yep, HDACs and HATs. Yeah. Okay, so enzymes that are going to help modify the histones. Yep. Yep. Can I get my ears checked? Good. Anything else? RNA polymerase. That's a big one. Yep. Okay, good. So what we're going to talk about is how the structure of the chromatin relates to its function. And so what this figure is showing is pretty much a summary of everything. I like it. So maybe after you finish studying this bit, you might want to come back to the slide and look at that figure and make sure it totally makes sense to you. Right, so it's talking about how you have your histones bound to your DNA, and when it's inactive, you might have your transcriptional activators or TATA binding protein, TATA binding protein, TATA box really, obscured by being tightly wound around histones. And how when you have activation of gene expression, you'll get recruitment of these chromatin remodeling complexes. These large subunits, complexes of multi-subunit proteins that are going to act to move your histones so you can gain access to these important binding sites in your DNA. And so then you'll get your binding sites for your activation and your TATA box and all your general transcription factors and all that. You'll get those available for binding, and then you can have assembly, so you'll have a transcriptional activator protein that will recruit your general transcription factors that will bind to your TATA box, which will bring in your RNA polymerase. And that's pretty much the gist of it, right? You guys have learned this before. We're just going to learn it slightly more deeply. Okay, so let's just start really simply here. So let me ask you, is the chromatin structure, so we're seeing two cells here, right? You've got your dividing cell. What's this cell doing other than dividing? What process is happening here? Anaphase. Yep, mitosis. Anaphase. And then this cell, where is this cell in the cell cycle? Interphase. Hey, Mike. But interphase, right? So is this chromatin structure same or different? So tell me what's different in these two cells?
The one on the left, the chromatin is more condensed. And the one on the right, it's not condensed.
So more condensed on the left than on the right. Yep. Anyone else? I saw you in the back. You're going to say the same thing? Does anyone want to add to that? Where do you think more gene expression might possibly, where can you bet that more gene expression might be going on? The left or the right? Where? I'm hearing the right. The right. Yep, so highly condensed chromatin, is it available for gene expression? Not so much. Now, is there no gene expression during cell division? I'm not going to say that, but I would say very, very little, if any. I'd say that highly condensed chromatin usually equals very little gene expression. So when you think about the function of a chromatin here, you can think about it in terms of multiple things, right? What kind of function is this chromatin playing here, right? So it's highly condensed, but what function does that serve? What would happen if it weren't condensed?
It's more labeled almost everywhere. You don't have space, so you just try to do.
So there's something about space. So it needs to be condensed so it can be compact. But what else? Yep. Okay. So your genome is packaged. So you're ready for cell division. What else? Yep. Right. So during cell division, your chromosomes need to be pulled apart and divided equally to daughter cells. And is it easy to divide that equally to daughter cells? This non-dividing cell? No, it's really hard, right? It's all tangled up like a big, it's like my daughter's hair. one of my daughters, she's like, I swear she does this while she's sleeping, because she has like a big interphase nucleus in the back of her head every morning. Yeah, so if I just try to rip her big knot apart, will she love me? No. I hope she'll still love me, but she'll be mad. That is for sure. But yeah, so if you tear apart your chromosomes to separate them into your daughter cells and they're not condensed, you'll probably tear apart your DNA. And you will not equally segregate to different cells, right? Okay, so that's one function. Yeah, so let's move on. That was fun. Any questions? I'm going to tell my daughter she has an interphase nucleus. She does what? Yeah.
I learned that it's called heterochromatin, whatever. Which of them is heterochromatin?
Well, heterochromatin, okay, so we're going to bring it up a little bit more. So you're talking about heterochromatin and euchromatin. Heterochromatin is highly condensed and compact. Yes. But I wouldn't really call condensed chromosomes during mitosis heterochromatin. Heterochromatin more has to do with an interphase, how you have some regions of your chromatin that are very densely packed and silenced. So, and euchromatin is your more open, transcriptionally active DNA. Yes?
Heterochromatin coming to bar bodies. Is that how you call it?
Heterochromatin and bar bodies. Yes, but I can't say anything else about that right now. Because that's your silenced X chromosome, right? Am I right? Bar body, silenced X chromosome? Right, so that will be, yeah, but we'll get back to that. Yeah. I think I have something more on heterochromatin and euchromatin coming up. Any other questions? Yeah.
I think I got confused. So the dividing cell has a condensed chromatin?
Condensed chromosomes, yeah. So they're condensed, they're tightly packed. And that's why you see these lines, right? That's like the chromatin, all those histones are wound up on each other and bound to each other. And so it's super tightly packed. Okay. So chromatin structure changes throughout the cell cycle based on whatever is happening at each point of the cell cycle. So in interphase, you're having gene expression and chromosome replication. Do you need heterochromatin, or do you need condensed chromatin, or do you need open, accessible chromatin during DNA replication and gene expression? Open. That kind of makes sense. You need all these enzymes and proteins to get in there. You're going to want things to be open. And then at mitosis, you have highly condensed chromosomes because you need them to be organized and prepared to be separated equally to daughter cells. And then you're going to divide and go back into interphase.
During meiosis, you're doing the crossing over.
Yes.
So at that time you have access to it.
So meiosis, think of it as another version of mitosis, right? Because you're still segregating. So the question is about what's going on in meiosis, and there you're still separating chromosomes, right? And so you need condensed chromosomes, right? Yes, when they're near each other, there will be that invasion, and you will have the crossing over. But I would still say it's condensed. You're making me question myself now, though. Okay. So here's your chromatin at mitosis versus interphase, right? So here are your highly condensed chromosomes ready to be segregated to daughter cells, and here is your completely open DNA. Yes.
So in interphase, we have like the more loosely packaged. So parts of it that are more compact, does that have to do with the genes that aren't currently being expressed?
Yes. So in interphase, when you have open some regions that are open. So you're not condensed chromosomes in interphase, right? And so you'll have some regions of your DNA that's heterochromatin, and it's not. These are genes that are silenced and not being expressed.
So it's variable, so if like something's being expressed, it can be other with you.
Right. So it's not. So don't. So heterochromatin. Okay. So you're asking whether it's kind of like a static state or whether it's dynamic
Right,
and changing.
right.
So there are regions of the genome that are always in heterochromatin because don't forget it's not one cell. Well, in our bodies it's not one cell. I guess we could talk these differently, but let's talk mammalian cells for a minute. So like compare
Liver cells versus skin cells.
liver cells versus skin cells, or I was thinking erythroid cells, cells that are going to become like red blood cells, right? So you need hemoglobin in those cells. Will you ever need to make hemoglobin in a skin cell? No. So you can safely assume that for the life of the skin cell, those genes are there with their presence. They're permanently silenced and present in heterochromatin. Make sense? Yeah. Okay. Did everyone catch that? Okay. Any other questions? So liver cells also would have a different type of genes that are going to be expressed that definitely would not be expressed. A lot of these enzymes. Yeah. Okay. So let's keep going. So you can imagine how if you try to separate this during mitosis, it's not going to be very neat, right? And so having this condensed form makes for easy chromosome segregation. Yes?
Is there a relationship between cell division and replication?
Is there a relationship between cell division and DNA replication? I'm going to ask someone in the class to answer that.
Well, you need to replicate your DNA in order to divide the cell, in order to copy the cell. So that's the greatest thing to do.
There you go. Right.
Because I was thinking about the brain cells which don't divide. So does that mean that DNA does not replicate?
Not okay. So she's talking about neurons that don't divide very frequently at all. Does that DNA replicate? Well, not as frequently as say skin cells, right?
But it still has all the machinery to replicate. It just doesn't.
Everything has the machinery, but you're just stuck in G0. Remember, you learned the cell. At some point, you learned cell cycle where there's, S phase, M phase, then you have this interphase, S phase, and then you're just going around that cycle over and over again. Copy your DNA, divide. Copy your DNA, divide, right? But sometimes you can go into G0 where you don't do anything, and that's where those cells tend to hang out more. Okay. Questions? I'm getting great questions today. Okay, let's see if we can keep moving here. Okay, so here's your condensed versus your decondensed chromatin. And so you can see you have your nucleosomes. Your nucleosomes are histones with DNA wrapped around. That's what our paper this week in recitation is about. Trying to understand how much DNA is wrapped around each histone, so the length of DNA per nucleosome. So in your condensed chromatin, you've got these nucleosomes are kind of packed up on top of each other. And so there's this higher ordered packing of your DNA, whereas in your decondensed chromatin, it's more of that beads on a string. So DNA wrapped around histone, DNA wrapped around histone. And so where are you going to have more access to transcription factor binding sites? Up here or down here? A or B? Right. So it's pretty clear just looking at the picture. Okay, so this is the paper that we're doing in recitation. It was really the discovery of the nucleosome. That was in 1974, where really people were asking the question of how much DNA is wrapped around each histone to make up your nucleosome. And so they unfolded their chromatin and got that kind of beads on a string decondensed chromatin, and they treated it with a nuclease to cleave it in between the histones. And then you get an individual nucleosome. So that's your histone with your DNA wrapped around it. And so then you can dissociate it into its subunits, which is your histone octamer and your DNA. And what they were doing in this paper is trying to understand how much DNA is wrapped around each histone. So here you're treating with a nuclease. What is a nuclease degrade? DNA. In this case, DNA. Did I see your hand go up?
Yeah, that was my question, that if the nucleus is digesting the chromatin, then is it pieces of DNA that is left behind or pieces of histone?
So nuclease digests nucleic acid. So now you can answer that question. What's left behind, your histones or your DNA, or both? Histones, but nucleases can only digest DNA that they have access to. I just made nuclease to a person. It can only digest DNA that it has access to. Remember, when you have an enzyme and a substrate, an enzyme needs to bind to the substrate sufficiently in order to carry out the enzymatic reaction. In this case, nuclease is an enzyme that needs to bind to DNA to degrade it. Now, can it access the DNA wrapped tightly around a histone? No. So what you end up with is your histones and that little bit of DNA wrapped around. So, and then what they did is they then they treated. So this is not straight out of the paper. This is kind of a summary of a few different studies that happened at the time. So here they actually are then treating the nucleosomes with high salt. So what is that doing? Why high salt?
Degrades the nuclease.
Degrades? Oh, that's interesting.
Denatures
So I just heard denatures the nuclease.
the enzyme.
Well, it would do that, but that's not the purpose. Yeah. Right, breaks the ionic bonds between what? What and what? Histones and DNA. So the histone subunits in the DNA, and so that's how it gets separated into all the pieces. That's one of those things I didn't really understand until graduate school, is how salt content truly affected things, and it's become very useful in doing research, because often when you're trying to analyze whether two proteins are interacting, you have to think about your salt content, because if your buffers that you're working with have high salt, will you ever find out if two proteins interact? No, because those ionic bonds will be killed by that high salt. Right. So that's a little intro. Sorry for those of you who had recitation without this. It's never good to be the first recitation. Usually I do try to integrate that stuff in, so you're not like, I don't know what recitation has to do with what we're learning. Any questions about this? Yes.
Like regularly we use this, the hat part to remove DNA from histones. In this case, are we using like the salt, same thing as the hat, or is it?
So you're asking about the comparison between HATs, histone acetyltransferase, and the function of the salt. Well, a HAT, in which we're going to talk about, the histone acetyltransferase is actually working within the cell, whereas this is happening in a tube. So, and the histone acetyltransferase is transferring an acetyl group to the lysines, which is loosening that histone DNA interaction. And I'll go through that a little bit more deeply shortly. Yeah, we'll get to it. But so in both cases, you are disrupting charge interactions between your negatively charged DNA and your positively charged lysines on your histone. In both cases, but one's more of an in vivo function, and the other is more of an in vitro assay or experimental method. Yeah. Any other questions? Okay, so here's the structure of your nucleosome core particle. There are all these, remember, there's histone H2A, H2B, H3, H4. So it's an octamer in the sense, did you have a question?
Yeah, I was gonna ask.
Should I go back?
Yeah. Does the salt concentration also break up the four parts of the histone, or does it only break off the main parts?
So it messes with any interactions that require charge because it's those ionic bonds that are keeping the positive, negative charge interactions that are allowing for your histone subunits to come together and for your histones to interact with your DNA. It's actually messing with both of them. Now you can get more specific and talk about how much salt you need to disrupt what interactions, because some things might bind more tightly than others, and therefore we need higher salt, but we won't do it. Okay, so you've got a side view. So the color is your histone subunits, and then you can see the gray is the DNA wrapped around it. And then this is, so this is more like, that's funny. So this they're calling the bottom view, and this they're calling the side view. But here you've got your DNA wrapped around it here, and then here's your DNA wrapped around it here. So that's your nucleosome. So the subunits are going to assemble into an octamer on the DNA. And so you'll have your H3 and H4 subunits that'll form a dimer. Dimer, what is a dimer? Two things binding to each other. And that dimer will form a tetramer, because remember it's an octamer, which means it has 8 parts. And so you're going to have your H3 and H4 dimer bind to another H3 and H4 dimer to form a tetramer, and it's going to assemble with your DNA. And then you have your H2A, H2B dimer, and 2 of those are going to form and bind here. So notice there's these N-terminal pieces sticking out. And so those are important. Now, do you need to memorize how the histone subunits associate with the DNA? Who thinks you should memorize that right away? Okay, good. Yeah, I will never ask you to regurgitate the assembly of histone subunits on DNA because that's not useful to me. It's important that I show it to you and that your mind is open to it, but you can look that up if you need to. I'm more interested in you understanding the function of things. So I do want you to kind of take note of these histone tails, okay? So most of the interactions of the histones with the DNA are with the backbone, so it's not hugely sequence specific. But some DNA sequences are preferred because if you think about it, the DNA is wrapping around and certain sequences are better for that compression that needs to happen. So your TA dinucleotides are preferred up against the histone. So that's that's your minor groove. And your major groove is your, well, basically, remember you learned that your T and your A only have 2 bonds and your G's and your C's have 3. So your T's and your A's are going to compress more readily and fold more easily. Okay, so remember I talked about these N-terminal tails, right? What do you think the purpose is of these tails? You've probably learned, you all took 309, most of you took 309. Yes?
You modify them to take the charge on, like make this more negative, so the unit's negative, so they start.
Okay, so you modify them. So I heard something about changing the charge. So there's modifications on tails and on core system. Yes? Mm-hmm. Right, so they're involved with recruitment of other proteins such as transcription factors. So they're modified. They recruit other proteins. What else? Anyone from the middle here? Yep.
The tails, when are acylated, acylated.
So one of the modifications that occurs on these tails is acetylation. Yep, so the acetylation and deacetylation of these tails affects what what's being recruited, and if you're talking about the core histone, affects the charge and interaction with DNA. So I know that's where you were going with that. Yep. How about over here? Yep. So some tails are involved with association with other nucleosomes, and that's involved with what? When nucleosomes bind to nucleosomes, is that forming? Huh? For condensation, for forming chromatin, for forming heterochromatin. Yep. So that's that higher order packing. What else? Anything else? You guys are pretty much getting it all. Okay, good. Right, so chromatin structure and function is regulated through post-translational modification of histones. And they can be modified in two different major places. One is on these histone tails, so this N-terminal tail of each of these histone subunits that's sticking out. And that often is involved with recruitment of other proteins or the higher order packing of the nucleosomes together. And then also you can have a modification on the core of the histone itself. And so what I'm showing you here is many types of modifications. There's methylation, acetylation, phosphorylation, ubiquination. There's probably more, I'm sure there's simulation. And they all kind of combine together to create this histone code. So it's kind of like a code that different proteins in the cell recognize and recruit different proteins to either reorganize your nucleosomes, recruit transcription factors, loosen the DNA, tighten the DNA, all of that. Okay, so I just talked about post-translational modification. What is that? What does it do? Yeah.
It alters the gene expression that happens in DNA.
Well, give me specific. Okay, so
For instance, methylation and silence are basically.
okay, so I'm hearing it alters gene expression. I want you to go super basic. Go back to Bio 201. What is post-translational modification? Not with histones, just in general. General. Yes.
Turning on and off proteins. Is that
Turning on and off. I want more
Determines
specific.
the function of the protein.
Determines the function of the protein. Yep.
It's a mechanism occurring after translation.
Some mechanism occurring after translation.
So it can modify the RNA, which would alter the protein.
So it's post-translational
Signaling.
modification. So you're actually altering a protein. But, yep. Altering the protein, yep.
Signal transduction.
Okay, there's a lot of post-translational modification in signal transduction. No one's hitting it quite on. So I'm hearing a lot of really great things, but I want more. Yes. So it affects protein folding?
It's the protein itself.
It does affect it. So when you talk about conformational changes that occur due to post-translational modification, yes, it can affect structure. You're getting there. Even more than that, yep. So you're modifying amino acids in the protein, so you're changing them in some way.
Or trafficking.
Trafficking. I know you're like
Trying.
topics. Okay, so yes. Everyone in who had 408 with me, I'm very disappointed. You guys should all be knowing. Yes. She shouted out ubiquitous. Very good. That is a post-translational modification. Yes. Okay. So signaling. Okay. So a lot of this. So post-translational modification affects everything. It affects structure. It affects function. It affects signaling. It affects gene expression. It affects all these things. But so the way I think of most post-translational modifications, though, is it is changing the shape of the protein. And it's affecting what it interacts with, which is affecting some sort of function. Not all the time, but a lot of things. Acetylation, methylation, what you're doing is you're actually changing the shape of the protein in that little region, which is determining whether or not a different type of protein will or will not be recruited. And that other protein is going to have a function, and that function might be recruiting some nucleosome remodeling complex, for example. So what am I talking about? I have no idea. Okay, no, I know exactly what I'm talking about. I love post-translational modifications. So specific post-translational modifications create and remove binding sites. You can think of it that way, especially here. With ubiquitin, it's a little different because often one of those binding sites is in association with the proteasome, which is going to result in that protein being degraded. But it also, there's ubiquitin that's also involved in recruiting other proteins. So you can think of if you've got a protein, and let me draw. I'm going to try to use the document camera more because I think over there I lose everyone here, and over here I lose everyone there. Okay, so if you have a protein that's shaped like that, like Pac-Man, and it normally interacts with this other protein, and the binding site is here, right? That's kind of like the region of interaction. Well, one form of regulation is often, well, thank you. One form of regulation is often adding a big phosphate group here. If you add a big phosphate group in the binding site, will that protein be able to bind anymore? No, because it needs that binding site between this part here and this part here. That's where your charge interactions are forming, right? And if you suddenly throw a phosphate group here, it's going to mess with it. So you can think about it's either preventing binding or it might activate binding because maybe there's another protein that has it has this SH2 domain and it wants to bind to that. And so that's a form of regulation. In some cases, protein A needs to bind to protein B. But in other cases, depending on the environment, some kinase is going to come along and phosphorylate right there, and that's going to prevent interaction with B, but it's going to recruit C. And that all has to do with the function of what's trying to happen in the cell. So that's the easiest way to think about, that's the easiest way for me to think about post-translational modifications, especially with this. So I said something just now. I said SH2 domain. And this is a perfect opportunity to introduce the concept of domains, because I'm going to talk about domains a little bit, and people always get really confused, and they think it's a specific protein or it's something having to do with kingdoms and phylogenetics, and no. In this case, it has to do with binding sites or regions of proteins that do certain things. So certain post-translational modifications, they create and they remove protein binding sites. So you can have phosphorylation on a tyrosine. So this tyrosine would be attached to a protein. And you'll get phosphorylated, and then in that case, it's going to recruit some protein that has a SH2 domain. So it has a region in that protein that likes to bind to phosphotyrosine. And so then there's bromodomains and chromodomains, and those are very much involved with gene expression and chromatin structure and function, and that's why I'm introducing this here. A bromodomain is a region in a protein that likes to bind to acetylated lysine. Right, so we talk about histone tails being acetylated. They're acetylated on lysine. Well, the reasoning behind that with the tails rather than the cores is that you're usually creating a binding site for a protein that contains a bromodomain. So this little blue thing is supposed to be a protein containing a bromodomain. And then you could also have methylated lysine, and that might create a different type of binding site for a chromodomain, for a protein containing a chromodomain. So the big domains to think about at this point, I bring in phosphorylation because that's the most common post-translational modification people have heard about up until now. But really, some big ones to think about are methylation and acetylation here, and in recruiting other proteins that will contain the appropriate domains, either a chromodomain or a bromodomain, depending on the modification. Questions about this concept?
I mean, I just don't get what the bromodomain is. So the protein contains bromodomain, but what is it?
It's a binding site. So the question is, what is a bromodomain? It is the part of the protein that binds to acetylated lysine on another protein. Yeah. So people like scientists like to name things, and bromodomains are called bromodomains because they have a structure in that protein that is similar in many proteins that bind to acetylated lysine. So there might be 100 different proteins, probably more, that contain bromodomains, and you can predict without a doubt that those proteins bind to acetylated lysines, likely associate with histone tails, and are probably involved in some way with gene expression or chromatin structure. Yeah.
So all of these domains, when these tyrosine lysine when they bind, that's altering the protein shape.
So when you have your modification, so when you have phosphorylation or acetylation or methylation, that's altering the protein's shape. And that's affecting what protein's going to be recruited and bind there. If you're methylated, you might recruit a protein containing a chromodomain. If you're acetylated, you might recruit a protein containing a bromodomain. If you're phosphorylated on a tyrosine, you might recruit a protein containing an SH2 domain. You could probably guess that receptors that are highly phosphorylated on tyrosines tend to recruit a lot of SH2 domain containing proteins. People always get confused with this, so sit on it, think about it, and bring it up again with me if you're confused still. Any other questions before I move on? I'm not really moving on here. We're going to kind of talk a little bit more about this. Okay, so protein domains, again, it's the part of a protein that does a certain job. It's not a modification. It's just a part of a protein. So I put this figure in because I was trying to find a figure that made it very clear. It's not showing up nicely at all. Hopefully it'll show up on Blackboard a little bit better. But so here it's just showing these different secondary structures. Here's an alpha helix, here's the beta sheath, who cares about that? Here's it showing a certain domain, which is really just a bit of protein structure kind of coming together and folding together. But then it's part of a protein. So here's the whole protein, and you can say that, let's see if I can do this better. You can imagine, let me use my arrow, that this part here that I'm outlining with my arrow is one domain, and it might have a job. Maybe that job is to bind ATP and hydrolyze it to ADP for whatever. This domain over here, this little bit, is a different domain. It's still connected to the rest of it. It might have a different job. Maybe it has a bromodomain and binds to acetylated lysine. And then if you were a researcher and you were studying functions of proteins, you can look and based on the known functions of these different domains, you can kind of predict the job of the protein. You say, oh, this has some ATPase domain and it has a bromodomain. Maybe it's a nucleosome remodeling complex or part of a nucleosome remodeling complex just because you work on that and you know about that. Okay? Okay. So you can think of proteins as modular structures. right? Over time, different structures have evolved to do different things really well. And so with each protein, it's not so much that you're creating this new mess. It's actually, you can think of it as there's these different structures that are taken and assembled together for the protein to do its job. So all these 1, 2, 3, 4 proteins all have bromodomains. And so you can guess they all associate with acetylated lysines. Bromodomains, so this is actually showing the structure of the bromodomain, and it's binding here to acetylated lysine. And so it has a similar structure and a similar function. The structure of protein has to do the function of the protein. So they're modular, okay? Okay, so what does all this domain and post-translational modification talk have to do with protein structure and function? I'm going to try to bring us back for a second. Right, that's because histone modification affects chromatin structure and function through increasing DNA accessibility or recruiting proteins that will affect structure and function. Okay, so understanding the concept of histone modification and its effects on chromatin structure and function is huge. This is like what this whole next exam is on. Ooh, did I just say exam on lecture free? I'm sorry. So it's an important thing to understand. So this figure is showing, again, your histones. I'm keeping you too long. It's showing your histones bound to your DNA, and each little dot is a different kind of modification. So there's a lot going on here. So we'll start here on whatever day is next. What was that, Thursday? Thursday. Please come see me if you still need to take a quiz or on Blackboard or whatever.